Good morning. I am Dr. S.SANTHALAKSHMI from DKM College for Women, Vellore. So in this session we are going to see about the basics of amino acids. Amino acids are the organic compounds which are containing functional groups such as amino group as well as the carboxyl group. So here we have a carboxylate and here we have an amino group. So they are the basics of proteins. So the basic unit of proteins are amino acids. Nearly 20 amino acids are found in proteins, and we have different classification based on the position of amino group in the amino acids. For example, if the amino group is present in the carbon next to carboxyl group, that is said to be alpha amino acid. And if it is present in the third carbon with respect to COOH, that is said to be beta amino acid, and next means it is said to be gamma amino acid. So depending upon the position of amino group, they are classified as alpha, beta and gamma. And also, so an example is given here. For example, glycine. In this glycine, the amino group is present in the alpha carbon, so it's an example for alpha amino acid. Similarly, in this, the beta propionic acid, amino group is present in the beta position of the compound, so this is an example for beta amino acid. So based on the charges, that is the number of amino groups and carboxyl groups present in the amino acid, they are classified into three categories. If an amino acid is containing equal number of amino group as well as the carboxyl group, they are neutral. So they are said to be neutral amino acids. If it is containing more number of amino groups than carboxyl group, they are example for basic amino acids. Similarly, when a compound is containing more number of carboxyl group than amino end, they are an example for acidic carboxyl groups. So this is acidic amino acid. And based on the nutritional requirements, we have two different amino acids. First one is essential amino acid, which is otherwise said to be indispensable. Another one is non-essential amino acid, which is otherwise said to be dispensable amino acid. Thus, essential amino acids are required for our body, but we cannot synthesize this, so they have to be provided to the body through diet. Example for this is valine, leucine, isoleucine, etc. Similarly, the amino acids which are synthesized by our body are said to be dispensable or non-essential. Example for this is alanine, cysteine, tyrosine, etc. So, how to name organic compound, that is amino acid? We can give different types of nomenclature for this. Either we can use a 3-letter word or we can use a single letter word. For example, in this we can see alanine can be represented by using 3-letter abbreviation called ALA, A-L-A, or we can give a 1-letter abbreviation like A. Similarly, arginine can be given as ARG. And it can be represented as R because we have already alanine, A is there. So this is represented as R. So asparagine can be represented as ASN or N. So like that we can give the nomenclature. So in this we can see the structures of amino acid. So here, for example, when you are taking this glycine is containing one carboxyl end and one amino end. So it is a neutral amino acid. For example, when you are taking this arginine, here we have 2 amino groups and 1 carboxyl group. So this is basic in nature. Similarly, when you are seeing this glutamic acid, this is containing 2 carboxyl ends, whereas we have only 1 amino group. So this is an example for acidic amino acid. When you are seeing the properties of this, all the amino acids are water soluble except glycine. All amino acids are optically active in nature, and the dipole moment for these amino acids are very high, and they are neither basic nor acidic in nature. An important thing regarding this amino acid is when they are dissolved in protic solvents like water, they are existing as a dipolar ion. So they are otherwise said to be Zwitter ion or ampholites. For example, when they are dissolved in water, so the structure will be like this. So it is existing as COO minus and we have NH3 plus also. So it is acting as both the proton donor as well as the proton acceptor. For example, this bitter ion can donate H plus ion and it can be converted into a cation, anion. Similarly, by accepting a proton, this is converted into a cation. So this is existing as a dipolar ion in protic solvents. Another important point regarding this amino acid is what is meant by isoelectric point. Isoelectric point means, for example, when you are taking this any particular amino acid in water and when you are passing EMF, depending upon the pH, they will be moving either towards cathode or anode. For example, when you are saying this in acidic medium, so this is a neutral one. In case of lower pH, the solution will be acidic in nature. So in that case, this amino acid is existing as a cation. So under the influence of electric field, these cations will be moving towards cathode. Whereas when you are increasing the pH and when the solution becomes alkaline, minus ion is existing, that is the solution is existing as an anion. So under the influence of electric field, the amino acid will be moving towards anode. But when you are adjusting the pH at one particular pH, the amino acid will not move either towards anode or towards cathode. That particular pH is called as isoelectric point, and this is very important for the separation of amino acids, and this will differ for amino acids. Thank you.